Hello, my name is Sishoya Duggan. I am a fellow of the 2023 cohort of Mark Fellows, and I am studying the origin of the reptile infecting adenovirus in the Geneva lab here at Rutgers Camden.
Hi, I'm Alexis Winters, and I'm currently a senior biology student in the Geneva lab here at Rutgers, and I have also been investigating squamate adenovirus. And today we will be presenting the phylogenetic analysis of adenovirus found in Anolis sagrii and Podarcis sicculus. While we worked separately on different projects, each in different stages of their completion, they both aim to further the knowledge of squamate adenoviruses. Adenoviruses, also known as ADVs, are DNA-dependent viruses that encrypt their own replication factors when entering a host. ADVs are commonly found in many species, each belonging to their own genus. Most reptilian cases are caused by ADVs within the adenovirus genus. ADV is highly present in the pet trade, but little is known about wild populations. To better understand wild populations, I investigated samples from the wild invasive Podarcis sicculus. We were sent 10 samples from regions of Philadelphia, Boston, and New York. After extracting DNA and performing the first nested PCR of these 30 samples, 13 are potentially positive for ADV.
In previous studies, most reptilian cases were caused by ADVs within the adenovirus genus. More recently, two individuals of Anolis sagrii from the island of Staniel Key were found to be infected with ADVs more closely related to the mast adenovirus genus, which was previously known to primarily infect mammals. In hopes of better understanding the origins of these novel ADVs from Staniel Key, I have continued ADV research with the various Anolis species that were collected across the West Indies. After extracting DNA from 42 preserved lizards and completing a series of nested PCR tests, sequences, and phylogenetic inferences using Mr. Bayes, I was able to find 8 new ADV strains from the islands of Staniel Key and Conception. Within these 8 ADV strains, 3 new evolutionary groups were identified. The group once assumed to belong in the mast adenovirus family was found to be sister to the Cia adenovirus family, which commonly infects frogs, tortoises, and birds. Another group of Staniel strains were found to be sister to an existing Bimini-based taxon. Prior to my sequencing, this taxon only included Bimini strains. The final evolutionary group originally consisted of 2 existing strains from Little Cayman, but was found to be sister taxon to one of my strains from Staniel Key. These results indicate that ADV has the potential to move between hosts. The presence of ADV strains outside of the adenovirus genus suggests this inference. In addition to this, the widespread findings of Staniel Key strains across the phylogeny answer the question of what strains are present on the island. Going forward, I plan on extracting samples from other islands not heavily represented on the phylogeny to better understand the various ADV strains present along with their relationships.
In the future, I plan to sequence the positive samples and then build a robust phylogeny to better understand how ADV affects Podarcis sicculus.